Welcome to Backstage at Upstage, a presentation of Upstage lung cancer, which uses the performing arts to raise awareness and funding for lung cancer research. Here's your host, the founder and president of Upstage Lung Cancer, Hildy Grossman.
Hi, I'm Hildy Grossman, and we're so excited to have you with us today backstage at Upstage. And here's my good pal, Jordan Rich.
Thank you, Hildy. We had such great feedback from our recent podcast on the Genome Project that we've invited our special guests back because there's just so much information we can impart. With us again today is Jane Wilkinson. Dr. Wilkinson is Senior Director of the Broad Genomics Alliance Management Team at MIT and Harvard. And also back is Assistant Biology Professor at the Koch Institute for Integrative Cancer Studies and Research at MIT, Dr. Francisco J. Sanchez-Revera, working hard in his laboratory to uncover the secrets of the genome and better ways to treat people with cancer. Hildy, you've put together another great podcast, so I'll let you take it away.
We're so happy to be back and to welcome Jane and Francisco, who helped create our fabulous podcast, What Did the Human Genome Project Ever Do for Us? Don't miss it. Take a look at all the podcast platforms and find it. You'll really enjoy it. We borrowed the idea for the title from Monty Python, but we really did explore this question. There are terms that are basic to genomics, including the name itself, as well as genes, DNA, sequencing, and oh, wait a minute, don't be frightened. If you didn't listen to our prior podcast, I promise as we talk about how the Human Genome Project shaped future efforts in genetic engineering and lung cancer, we'll explain these terms. It'll be ready for a first grader. So grab your first grader so they can listen as well. So Jane, who was a part of the original Human Genome Project, can you give us some idea about how the knowledge we gained from this project led to important testing we now do in cancer research?
Yes, absolutely, and it's so good to be back again. And it's really lovely for us to kind of really round out how we have moved from discovering the human genome to really putting it to use. I'm going to touch a little bit about testing, but for me, Francisco is the hero of this podcast because he's really going to tell us about how we've pushed this now into a tool. But anyway, genetic testing in cancer testing has been around for quite a while. And I think some of the most obvious applications of what the genome revealed to us and what has presented us now with testing options. I think there's one thing we have to talk about, which is there's always a hereditary component to cancer. So, for example, there are about more than 50 different syndromes that are related to cancer that are hereditary, meaning that you would have inherited a copy of the gene or two from your parents. Most of the hereditary cancer syndromes are associated with actually getting cancer itself, but there's also several genetic mutations that can be associated with increasing the risk of getting cancer. So, it's really important to understand the capability and the utility of those testing. One thing I will say, there's a lot of direct-to-consumer testing out there. You can jump on the internet, you can order a kit, and you can get your results back. I do think that that's incredibly powerful, and it really puts the tool into the hand of the patient. But I would also really, really strongly recommend folks who are thinking about using these types of testings around the hereditary component of cancer to really work closely with a genetic counselor who can really kind of help you walk through the risk and the outcomes and also the data itself. But I think, one of the most important things about understanding the genetics of cancer is actually doing genetic analysis on the tumor itself. And I think that's a tool that has really become very, very powerful in the clinic. And it's quite honestly now just become a standard, part of cancer care, which is just amazing, especially for someone who was fumbling around in the Human Genome Project, wondering where we would ever end up. This is incredibly powerful. So there's a couple of different tests that oncologists will run on the tumor itself. And the genetic testing of the tumor isn't necessarily on a solid tumor. It's not on a physical tumor, such as what we would expect to see in something like lung or breast cancer. There's also capabilities to do what we call liquid tumor testing. So understanding the genetic component of more bloodborne types of cancers. And I think what that kind of genetic testing does for us on the tumor and also on the blood itself is it gives us an idea of whether or not there is evidence of drug resistance in that particular type of tumor, and also on the kind of good news side of it, drug response as well. So by characterizing the tumor itself, we can really start to understand a menu of options for treatments for patients. And to take that leap into lung cancer specifically, there's a bunch of different genes that we're particularly interested in, EGFR, KRAS and ALK. Those are just 3 of many different genes that are implicated in lung cancer. And, we often use the understanding of the mutations in those genes in lung cancer to really decide what the treatment path is for patients, but then also critically important, and especially for folks who are listening to this podcast, because I know they are, we have some listeners who are really, really, really invested in this. Understanding the genetics of the tumor itself can also guide and steer patients into enrolling into clinical trials. I'm not saying that there's an answer to everything yet, but by understanding the characteristics of the tumor, especially at the genetic level, it does allow patients to enroll into clinical trials and allow them to be potentially part of the future of curing lung cancer. So, for me, that's the power of genetics when we're in it, when we're actually in the tumor and when we're in cancer itself.
I was going to say we've done at least 3, I think 4 podcasts looking at when you're talking about genetic testing of the tumor. Many people know this as biomarker testing. So that is exactly, looking to see if a person who is diagnosed with lung cancer, for example, has one of these biomarkers, such as you mentioned, EGFR, KRAS, and ALK positive. So those biomarker testings have now become, there's so many that can be tested for. There's some incredible companies that are looking at solid tumors and liquid tumors. So getting that information, I think the great thing is it will take it outside of the box. So we always think about lung cancer, we think about rectal cancer, we think about breast cancer, but it may be that your particular biomarker has more in common with a different organ than within the same organ. So it may be that you wind up getting medication that was aimed for example, like breast cancer, that may be more effective for another organ system's cancer. So it's just, we're just at the beginning of like uncovering all this wonderful information that's come from the Human Genome Project. It's just, it's wonderful.
There's another opportunity on the horizon as well, where folks are going to start looking at circulating tumor cells. So as tumors exist and grow within the human body, they shed cells as they're going about their daily business. And there are some opportunities to identify these cells that are floating around the body as a way to think about early detection and screening, but then also on disease progression itself. And the technology is advancing pretty quickly to the point where I think we're getting close to actually identifying the primary tumor when we pick up on a circulating tumor. which kind of helps to pinpoint where the tumor of origin is going to be. There's still a lot of work to be done in that field, but from a testing point of view, I think that's also on the horizon. I really want to hear from Francisco about how he's using the genome and how it is really shaping the future of cancer in his lab.
Can I just ask a question? And it may be that Francisco is the one to answer it. So what's the big deal about figuring out what the primary tumor is? Like, why is that important?
I think it's important to know the source of the circulating tumor cells, because it allows you to then, on one hand, guide treatment to an extent, knowing that the tumor, say the primary tumor, is in the lung or the pancreas or the liver or somewhere else. There's some therapies that could be tailored depending on the tissue of origin, independent of mutation. So that's one important point. The second one is that if a disease is metastasizing and you know it's derived from a primary pancreatic tumor, for instance, then that is much more deadly than other types of tumors that shed cells as they progress, because some tumors are much more metastatic than others.
That would be a clear path to a different kind of treatment. Is that what you're saying?
To an extent, but it's not, I'm not a clinician, so I can't really comment on the specific treatment paradigms, but to an extent, there is an ability to match certain therapies if you know what the primary tumor is. Yes.
That's great. I didn't want to divert you from Jane's question about what's happening in your lab.
Yeah, no,
So
I think that I first want to mention a couple of points on what Jane nicely set up the stage here. A few things. I think Hildy mentioned that there are certain therapies that are designed for some mutations in some tissues, but not necessarily others. But I think that's where the power of genetics comes in. For instance, knowing that two different tumor types have the same type of oncogenic driver by itself is useful information to suggest that a therapy targeting that driver might be useful in both of these tumor types, even though they arise from different tissues.
So what is an oncogenic driver? What does that mean?
Yeah,
for the first graders in the audience.
Yeah, so as I mentioned in the last podcast, genes encode proteins, and there are two main classes of genes that are involved in cancer, that drive cancer and those that prevent cancer. The ones that drive cancer are typically called oncogenes. KRAS, EGFR, and ALK are oncogenes. So when they get mutated or hyperactivated, they promote cancer. Those that promote cancer and those that inhibit cancer. Those that promote cancer are typically referred to as oncogenes, and those that prevent or suppress cancer are called tumor suppressor genes. So for instance, the genes that Jane mentioned earlier, KRAS, EGFR, ALK, those are oncogenes. So when mutated, they promote tumor development, progression, and in some cases, modulate the responses to chemotherapies and targeted therapies. But on the other hand, genes like 53, which I think we discussed briefly on the last podcast, but it's one of the genes that I study, is a tumor suppressor gene. So when mutated, it leads to cancer because that mutation inactivates the function of the gene.
I've got a question for both of our wonderful panel members, and welcome back. It almost feels like a very complex Sudoku game or crossword puzzle in that you now have outlined the mapping, which is so important. You've also outlined the two genes that bounce off each other and battle each other in some cases. Do you see the technology and the computer speed and all the important things coming along to make it easier to solve the puzzles? Because these are very tricky issues right now.
That's where genetics and genomics comes in. For instance, just to put it in perspective, the human genome defined the blueprint of what makes us humans at the DNA level. And only by knowing how the normal genome looks, how the normal DNA looks and is written, knowing that normal blueprint, is how we can know when things go wrong in terms of mutations. You can identify exactly what those mutations are because you have a reference to compare it to. So, for instance, genes like KRAS are typically mutated through a single nucleotide alteration. So one letter in the gene and in the genome is altered, and that is sufficient to cause the activation of this gene, an oncogene, and lead to tumor development and progression.
It's amazing. It's
Okay,
amazing.
just wanted to make sure that that was clear.
I think, yeah, no, you made it very clear. And for anybody who just wants a little bit more explanation, please do tune into our other podcasts where you did explain this in more detail. But I think you've said enough that makes it, possible to comprehend.
Yeah, so here is where the patient heterogeneity and diversity comes into play. You might have in the clinic 5 different patients, all of which have lung cancer. 2 of them might have one KRAS mutation, one letter changed, which leads to one particular KRAS mutation. And the 3 other patients might have a different KRAS mutation. Sounds simple, and you can say, well, all 5 patients have a KRAS mutation, therefore they should be sensitive to the same types of therapies. They actually aren't.
So are you saying that a KRAS mutation, what that means is that just one letter in the sequence is off? So it just depends on which one of those letters is off to
Exactly.
suggest it's a subtype or a different kind of KRAS. Is that right?
Exactly. And to make matters more interesting, but also more complex, some of these mutations that are different happen in the same location. The most common mutation is at position 12 of KRAS, and you can have various flavors of that mutation at position 12. Some of the most common ones are G12D, G12V, and G12C. And what makes this very exciting, but also complicated, is that there are now drugs that are able to directly target the G12C mutation, but there are not drugs, at least not in the clinic right now, that can target the other types of mutations. So by knowing the exact letter change, you can tailor a therapy to a patient.
Amazing. It's just...
KRAS, because it's the most commonly studied oncogene, but we have 19,999 roughly other genes in the genome that can be mutated in different ways in cancer. And so we basically have a very poor understanding of what types of mutations can alter the function of all of these genes. And to make matters more complicated, different patients have different mutations in all of these different genes.
So do they just set up these like extremely complex algorithms to try to pinpoint some of these variations?
So there's a combination of just knowing the sequence, knowing the mutation, just reading that blueprint for that gene, combined with years, decades of functional studies in the lab. So functional studies meaning there have been a variety of experiments in cells and animals and human clinical trials that have pinpointed the function of some of these mutations in cancer. So we know the function of some of those that gets integrated into some of the diagnostic and clinical treatment decisions nowadays, which is still, again, very impressive to me. And this is what we typically refer to as precision medicine or precision oncology. There are also computational methods that take into account protein evolution and conservation of sequences. I could get into the weeds if you want to, but I think just take my word for it that it allows you to predict to some extent what the most likely effect of a given mutation might be, depending on the gene and depending on the location. And then on the other hand, which is what, so in my lab, we combine some of those methods with functional genomics and genome editing, which I briefly touched upon on the last podcast.
I think it's really important for us to highlight that a lot of this work and a lot of this analysis around the implications of these the genomes in these tumors is really around using machine learning and artificial intelligence and deep learning, really kind of using tremendous computational powers to really interrogate and analyze the sheer amount of data that's just tumbling out of not just the human body in understanding the genetic characterization of the tumors itself, but also pouring out of the labs and out of the experiments and out of the research, which is really enabling us to kind of really understand what's happening at that cellular and genetic level.
We'll get back to the podcast in just a moment. As thousands of audience members know, Upstage lung cancer events, the concerts, are fun, meaningful, inspiring, and memorable. And you should know that we invest in cutting-edge diagnostic research to find lung cancer early and greatly improve on the 5-year survival rate. We also bring voice to the fact that young people get lung cancer. They really do. Unfortunately, doctors don't know how or why. Proceeds from our concerts support research to help find answers to these questions. Hildy Grossman and her team aim to entertain and inform, because the show must go on. Find out how you can help at upstagelungcancer.org. Now back to the podcast. Here's Hildy.
Upstage lung cancer is very, very interested in early detection. So I don't know that you've exhausted everything you wanted to say, Francisco. I'm guessing not. But is there, from all this complexity and understanding, what a normal, quote unquote, normal genetic profile looks like, is there some way that one could take this into the future to do some testing that doesn't cost an arm and a leg or is not too impossible to help predict or detect lung cancer, let's say, early?
There's multiple options there. One that involves imaging. Early imaging, I could see that becoming to some extent routine when combined with machine learning and artificial intelligence algorithms. For instance, this has been done in breast cancer, and there's a researcher here at the KI and also at the computer sciences department called Regina Barzilai, who has pioneered imaging-based machine learning methods to detect breast cancer, which could also be applied to other tumor types. So I could see that imaging becoming an important modality in that sense. The other one to me is really sequencing, DNA sequencing, because thanks to the Human Genome, Cancer Genome Atlas, and many other types of large-scale efforts, that has made the technologies and the sequencing costs drop dramatically. So sequencing is becoming more and more cheap every day. We right now, like I've said before, a patient goes into the clinic and their tumors are sequenced routinely, not everywhere, but in many of the cancer hospitals. A sequencing test, I don't know exactly the cost, but in terms of the sequencing itself, it gets cheaper and cheaper over time. So I think sequencing is probably our most powerful weapon in that sense, because if you can have sufficient genomic data to allow you to know whether a certain mutation that you previously didn't know if it's functional or not, if you know that mutation is there, you can probably use it to predict whether a person will develop cancer or other diseases in the long term.
So would that be easier to do an early detection using blood rather than, like a CT scan if you're talking about screening?
I think so.
I mean, I'm always interested in making it more available to the masses. Set up a CT scan. There are already so many criteria that you have to satisfy
Yeah.
to get that
So I mean,
done.
for instance, you can isolate blood from people, 100, 1,000, 10,000 people, and all of them can be sequenced. That is much faster than CTing or imaging all of those people. Some of the ethical considerations that we discussed on the last podcast, which is a completely different theme, but that's something to keep in mind. But technologies like 23andMe and others are doing something similar routinely, and I think based on the fact that these sequencing technologies are so cheap, I could definitely envision this becoming routine in the not so distant future.
And just a reminder for our listeners on the evolution of the cost of sequencing a human genome. The Human Genome Project that I was involved in cost $3 billion. Actually, it cost $2.7 billion, just for the purists out there. We can now think about sequencing a human genome for less than a couple of $100. That price drop is incredible.
And there are now new projects that are aiming to diversify the collection of human genomes that are sequenced. There's a project called 1,000 Genomes, and then there are other consortia that are sequencing genomes more completely from telomere to telomere. That's the Telomere to Telomere Consortium.
Wait, what's a telomere?
Telomeres are basically the end structure of the chromosomes that solve one of the biggest problems in molecular biology called the end replication problem. Basically, if we didn't have telomeres, our chromosomes would get shorter and shorter over time. But because we have those telomeres, telomeric proteins, they keep extending the ends of the chromosome so that we retain a complete and capped chromosome from end to end.
So they stay at a fixed distance or length?
Length, yeah. But that gets complicated, and there's some debate as to what the length of telomeres means, and but definitely implications in cancer in that sense, and they go in both directions depending on the tumor type and on the research. So yeah, our collection of genomes is increasing, is importantly becoming more diverse. So the initial human genome was just one genome out of the billions of genomes that exist in the world. Now new projects are diversifying that. And by that, I mean sequencing genomes of people from all over the world so that we have a better understanding of how genomic variation influence disease. And this relates to cancer, of course, but also many other diseases.
So, for example, what's the benefit of having the heterogeneity that you would get from sequencing or random sampling sequences from people all over the world versus, let's say there's a country, I know like Finland years ago didn't have much influx or exodus, so that you have a population that's pretty stable. What's the benefit of a more homogeneous versus heterogeneous population?
Icelandic populations and Finnish populations, those types of genetic and genomic studies are useful because you can essentially look at genetic crosses that you would typically do in the lab. And because most of them have a large percentage of their genomes is identical, you can find the consequences in disease development when you have two copies of the same gene. So each gene has two alleles, one from mother, one from father. And if you have the same two alleles of a given version or configuration or mutation, then you can assess what happens when you have those two alleles versus just one from one population and one from a different population. It allows you to infer the function of that gene in disease, but also the function of that specific variant that exists in that population. That means that if you only study that population, you're only getting information about that population. And there's substantial evidence over the years that different populations have different SNPs, single nucleotide polymorphisms. And those are essentially...
easy for you to say.
These are single nucleotide polymorphisms. That means that single letters in the DNA are different depending on the population.
So some populations wouldn't have the
So,
same letters or the
yeah, so you
sequence
might
of
have an
letters.
A, you might have an A in a gene, a position in a gene. I might have a T, Jane might have a G, and Jordan might have a C, for instance.
Yes.
So that is actually very important because it's been shown in over the years that depending on the type of SNPs that someone has, their propensity for certain diseases like cancer can be different. And this relates to, you know, disparities in both research, cancer research, but also in general across multiple diseases. So we need to understand how genetic variation influences disease development and treatment, not only for one population.
So wow, this is just it's mind-boggling, the complexity, the details, the micro data. I don't know if there's such a word, but anyway, the that comes from all this kind of research. I know Jane at the Koch Institute is doing cancer research on all kinds of issues and areas. So yeah, it's like how to take things at a microscopic level and then look at how applications can be generated, how detection can be created and thought about in an interesting, creative way. And then, yeah, so it's just, it boggles my mind what goes on in laboratories like yours and all over the world.
Yeah, so before I forget, just from our point of view in the lab, but we're one of the missions that we have is to assess the function of all these different genetic variants that are observed in cancer patients. So I'm interested in finding out when you get the DNA profile of a tumor from a patient, if we see 50 different mutations, what are the function of all those mutations and how those influence the development and therapeutic responses of those tumors? Because it's becoming more clear over time that every patient is unique. So understanding how the mutations in one patient contributed to that tumor, to that disease, and how it influences the treatment might not necessarily be the same mechanisms that are seen in another patient. We need to understand both what each of these mutations, each of these genetic variants is doing in the context of cancer, but also how the combination of variants in a given tumor sums up to influence that disease. And that might be different depending on the patients. G12D versus G12C in KRAS is just one example, and we need many more functionally validated examples of that.
Maybe this is our time to wrap up. Although every time we end, this is now the second time we've ended, I want more and more. It's so complicated and so interesting. I was just thinking as you were just, you know, Francisco, as you were just saying that last comment, what I've known or have learned is that in all of us, cancers come and go. And I think we may have touched on this last time, but people get cancers that come and go all the time. Some stay, some disappear. So I think part of what you were talking about is trying to figure out what process and what's involved in the ones that come and then disappear versus become active and dangerous.
Just as a last point, if you want to include it, that's where all the genome editing technologies come in, what I typically refer to as precision genome editing technologies, because using these technologies that are based on tools like CRISPR, we can engineer very specifically each of the mutations that are seen in cancer patients using experimental models in the lab to try to assess the function of each of these variants alone and in combination across different tumor types. Two different tumors might have the same genetic alteration, but that doesn't mean that both of them will respond to that same drug that targets that alteration. And understanding why that is important. We think it has to do with the both the combination of genetic events that are seen in that tumor and how they cooperate with the main mutation that is targetable, but also how the environment within the tumor and other types of variables influence those processes.
Obviously, we need another gathering. Like this is looking at lung cancer, asking questions about what CRISPR can do for us in lung cancer. I just think we have many more things to talk about. So I'm sure this wonderful gathering will continue again. I'm not sure we will have exhausted all the questions people might have about how we learn about cancer and its process and everything else, but this was wonderful. Jane, do you have a wrap-up comment for us?
We have opened up another can of worms and another direction to go into, and especially around Francisco's work around the genome editing tools. I really, really hope that we can spend some time talking about that next time if Francisco will allow us.
Because there are so many moving parts that relate to how this technology works.
Yeah, that sounds good to me.
Terms.
Yeah. We need the Francisco show.
We do. Well, that'll be, yes, Francisco, away we go. Many thanks. We'll see you soon. And Jordan, as always, please, everyone listening to this, listen to our other podcast as well. And you are going to wow people at holiday cocktail parties with your knowledge of genetics. So goodbye, stay healthy, and we'll see you soon.
To find out how you can join Upstage Lung Cancer in raising awareness and funding to beat lung cancer, visit our website, upstagelungcancer.org. We invite you to subscribe and download our podcast, available on all platforms. And we love reviews and ratings. After all, we're showbiz people. There's more entertainment and inspiration to come on the next podcast episode of Backstage at Upstage.